right, so we will start with our first speaker for today on the session of Healthy Oceans. It's my pleasure to introduce you to Erin Easton, Dr. Erin Easton from University of Texas, Rio Grande Valley. We'll be talking about improving scientific understanding of the Gulf of Mexico mesophotic octocorals to maintain healthy oceans. I will be standing off at 2 minutes before the 13-minute limit for timing purposes, and then you will be able to ask questions after that.
So I think 25 minutes, right? If not,
He wants to reflect there, but if it's 25, that's fine.
Okay.
I think they were 25.
Okay. Just making sure, because if not, I'll have to flip through some stuff I added.
Thanks for letting me go.
All right, so yeah, as Dr. Carlson mentioned, I'm talking about improving our scientific understanding of the mesophotic octocorals in the Gulf of Mexico. So much of my talk is more background and why it's really important that we improve our scientific understanding, and I have some summary of some of our preliminary results at the end. This is a very collaborative work, so I have all the acknowledgements at the end. And
Trouble. You guys are glad it's not turn it on.
okay, on first, thank you. There we go, that was the problem, thank you. All right, so Healthy Oceans, they provide a number of services. Among them is that they are very economically important. They provide a lot of economic services, and that is through fisheries, tourism, also in transport. Roughly 1/6 of all the jobs in the United States are related to the ocean in one of these three areas. They also provide us with resources from food to medicine to technological compounds. So if we don't have a healthy ocean, we're damaging our economy. And so that can be through fisheries closures, beach closures, and even damage to our infrastructure. And we're going to have reduced resources, so lower food quality and quantity, fewer medicinal resources, and technological compounds. So it's really important that we maintain these healthy oceans, and the way that we're moving towards doing so is through ecosystem-based management. And to do ecosystem-based management, you need to have these integrated ecosystem assessments that bring in high-quality data and indicators from a variety of components. So your climate, economy, culture, ecosystem, habitat, your stocks. And what I've really been focusing on are biodiversity, ecology, and function. In my study area, for the purposes of this talk, is the northern Gulf of Mexico mesophotic reefs. They're from a roughly 30 meters depth to 150 meters depth. And there's three reef complexes in the Gulf of Mexico via the South Texas Banks, the Salt Diapir Banks, and the Pinnacles and West Florida reefs. And those are indicated by, each reef is indicated by one of the grayer black circles. So these mesophotic reefs, they provide substantial amounts of topography and substrate for organisms compared to the relatively flat, muddy shelves. They also support habitat-forming corals and sponges. My interest is primarily in the corals, but both these corals and sponges in turn provide habitat. And so these reefs provide habitat for these unique faunal assemblages compared to their shallow and deep water counterparts and the surrounding muddy seafloor. They also provide critical habitat for commercial fisheries. That could be in terms of nursery or feeding grounds. But yet they're understudied relative to their shallow and deep water counterparts. So we know very little about their biodiversity, ecology, and function. So when we start studying these, there's some fundamental ecological and biodiversity questions we need to first start with, and that's who's who and who's where. So I'm again working primarily octocorals. This is my primary interest, although there are other groups and my collaborators are focusing on those. And then who's where, one of the reasons we want to understand that is so we can have habitat suitability models. So what are some of the tools that we use? Well, we use morphology and genetic tools. So we're doing morphological studies. We might be using in situ images. So we might just be surveying these reefs with video. And so we need to be able to accurately identify colonies, species from video. If we have a sample in hand, we're going to use microscopies. We can use macroscopic or microscopic characters. In terms of genetics, we often use barcodes, either for the nuclear or mitochondrial genomes. Those are short DNA sequences that we target. You can use next generation sequencing data, which can help you get at some of those functional questions, as well as biodiversity and ecology. But those are more they're more expensive to conduct, and there isn't quite the data set available. And then you can use environmental DNA as well when you're taking water samples and targeting usually a barcode region to amplify and look at what organisms are in the area where you collected that water sample. But when we're doing these assessments, we have a major problem, and that's with octocorals, inaccurate identifications or assignments are really common. And this is in part because we have no best practices. We haven't standardized or centralized the data that we would use, the criteria to assign individual species. It's often not up to date, so we're using different technologies now than we used to. The methods might not be suited to the survey methods we're currently using today. And another big one is your expertise or your experience of your observer or the person doing the assigning is going to differ among people. So just for some examples, in terms of morphology-based biodiversity assessment, it's further complicated by the fact that these diagnostic characters are not always visible in situ images. So if you're doing a video survey, we look here at this image on your right, there's numerous coral colonies there, most of which are octocorals. And in this image, with the video quality data that we have, there's probably only 3 to 5 colonies that I could confidently assign to species or even genus level. The vast majority we have to leave at family level. And that's just in part because we also don't understand we have limited data on the inter and intra-specific morphological variation. And if we look at an example where we actually have some samples at hand that were collected at some of these same banks, we here in yellow is the ultimate ID that we've given at this point. It's been assigned to the genus Thracis. And that's using both morphological data and genetic data. But if we look at the original field IDs, even when you have it in their hand, not a single one of them assigned it to this genus. Every single sample we have was assigned to one of 3 other genera. To the family level, or in some cases they recognized it was potentially a distinct species, but they didn't know what it was, so they gave it a code. Now, if we're using genetics, we have some other issues. So the barcodes that we use, because we use these short primers, they're meant to lay down on your target region and amplify. Sometimes they don't lay down there, so they can fail to work. And there's a variety of reasons for that, but they might fail. Or when the sequence comes back, there might not be sufficient genetic variation. And so here's an example of insufficient genetic variation for one of the target genes. When you're looking at these figures, what's really important is if you see a colored bar amongst the gray, where it's gray, they're identical, but they differ where I see one colored bar in these 2 grays. So if you're looking at the 2 species with the gray bars, and we have 2 differences, that equates to roughly 0.2% divergence between these 2 species. But if we collect more genetic data, say over the complete mitochondrial genome, they have almost a 10-fold increase in the amount of divergence. So there's much more data there for us to help distinguish these species. So we need to make sure we're using the appropriate genetic markers. In terms of eDNA, we have limited reference data. So this would be like going to a store and they have a bin of cans that all you can scan is a barcode on it. You no longer see the label. You take it to your little scanning machine, you say, hey, what is in this can? But in their database, in their spreadsheet, there is no data there, or the only data that's there is saying it's a vegetable. Well, that's great, it's a vegetable, but which vegetable, right? So that's often what's happening with these eDNA surveys. They would say, oh, it's a coral. Great, or it's in this family of corals. Great, but what species is it? So we're gonna be doing these biodiversity assessments. We need accurate reference data. And then there's also, in terms of next generation sequencing data, a number of limitations that I don't really have time to go into today. So let's talk about what are the downstream consequences of having inaccurate biodiversity assessments. Let's start with a more simplified scenario where we have a mesophotic reef from 50 to 150 meters depth, and we have colonies of different colors. So we have yellow, red, purple, and we're going to say based off of the descriptions that are available, we know, or we think we know, that we can distinguish these in the video survey based on color alone. yes, this is overly simplified, but shockingly, it's not too different from the reality. And so we're going to conclude that there's 3 species and 3 genera based on this. But once we have samples in hand, we do more thorough assessments with better reference data. We look and we find that there's actually 5 species and 4 genera. So our assessment, our understanding of this ecosystem has now changed. We have a biodiversity of 3 versus 5, right? So our alpha diversity is much higher in reality. We also know that then color is a diagnostic for some species, right? Some species can be yellow or red, and several species can have yellow colonies, for example. So what if we want to go further in terms of ecosystem-based management? We want to model where we might want to find these, where we might find these corals. So again, we have this simplified system at 50 to 150 meters. In this case, I've simplified it to only 2 colors, and our initial conclusion would be that we have 2 species. The purple species is found at depth shallower than 80 meters, and the yellow is found at depth greater than 80 meters. So again, we get that additional data, and we reevaluate, and we see that yes, we do have 2 species and 2 genera. So the biodiversity is the same. However, the one species is indicated by these small yellow colonies, and so it's found at depths greater than 100 meters. So it's found at depths deeper than we thought, and not quite as shallow. The other species is indicated by the larger colonies in yellow or purple, and we see that their bathymetric distribution is from 50 to 150 meters depth. So now we have our model outputs are going to be different because they're going to have larger bathymetric ranges or narrower bathymetric ranges. So what about when we have disturbance? How are results and our ability to respond and restore going to change? Well, again, same simplified model as the previous slide. We have 2 species and 2 genera. After the disturbance, based on our initial conclusions, solely based on color, we find that there's 100% mortality with the purple colony and 50% mortality of the yellow colony. But in reality, we look again, and what we found is that there's 100% mortality with one species, that indicated by the larger colonies, and 50% and 0% mortality with the other species. So then your questions that you might address are going to be different. You know, in the first scenario, you might have thought maybe it's just size or size alone that's indicative, or that one species is more susceptible than the other, but they're both susceptible. In the second, we find that one isn't susceptible at all. What does it have to do with the size? Does it have to do with the species? And so the questions we're going to address and the methods we use to do restoration are going to change. So in terms of what we've been doing to try and solve some of these problems are these 3 solutions. And so our ideal outcome is to improve our accuracy and consistency in terms of species assignment so that we can better understand these ecosystems and have better outputs and better restoration plays. So the first step is to collect more data. So there are roughly 75 known octocoral species on these mesophotic reefs. The vast majority of those had absolutely 0 genetic data available when I started this project. But when we do so, we need to consider the limitations of the methods that are being used. Also, we need to consider inequities. We're shifting towards newer technologies, but there's some institutions and states that cannot use that same technology. So we need to make sure that we're still producing data that can be comparable to data that all institutions are collecting. We need to compile that information and make it publicly available. And my pet project is we really need to have a good field guide so that anyone can produce good identifications. And so we don't have this problem of having a dozen samples, none of which are assigned to the correct species. All right, so in terms of the accomplishments that we have made in this work, is of those roughly 75 known octocoral species, we did some next generation sequencing methods that we can recover the traditional barcode as well as some of these other markers, including the complete mitochondrial genomes. And so we're at roughly 1/3 of those species we now have complete mitochondrial genomes for. We have all of those plus several additional species. We're nearing about 50%, for which we've obtained traditional barcode reference data. We've designed, to do so, we've had to design dozens of new primers. So when you go to amplify those regions, one of the reasons octocorals have been problematic to work with is that these traditional primers that are being used don't work for a lot of the species. So we had to work on designing new primers that would increase the success rate. So now they work, I don't know, about 90% of the samples I tried, they work for now. We also need to obtain that microscopic data. So we need to do microscopic analysis of all the octocorals because many of them were described decades ago before we were using more modern techniques. So scanning electron microscopy, light microscopy, we need to collect that data so it's available. Let's see. Yeah, and also in doing so, we've identified numerous taxa for which new taxonomic revision and several new species that have never been reported for the Gulf of Mexico before. And so just one example of what we found is going back to the sclerosis species. 2, here, the yellow colony, and the sclerosis species 1, the red colony. What you need to know about what's known about sclerosis, this genus coral and the Gulf of Mexico, or just in general, there's only 2 described species in this genus. One of which has very spindly, long sclerites. Those are the skeletal elements that are in their exoskeleton. It's a little difficult to tell with the lighting, but you can actually see them here. They look like grainy. The other species of sclerosis that's described is plate-like sclerites. So they're quite distinct, and if you have it in your hand, you can see that with the naked eye. So you don't even need a microscope to see that diagnostic character. So we also obtain genetic data for these, and again, everywhere the gray bar, the 2 species are identical, and everywhere there's a difference. This is just a small fragment of the complete mitochondrial genome. What we're seeing is that within a species, we're finding less than 0.1% divergence, but you're looking at roughly 2% divergence between the 2 species. So this is on par with what you would find divergence values for congeners or different species, and also within species divergence using these markers. So the genetic data suggests that they're different species. There's also some morphological differences in terms of their morphology. The placement of the polyps, they're much more like similar throughout, like they tend to come in pairs, but they're a little more random here. On the species, these colonies, the branches also connect to one another, but you don't see that in the yellow colony. So there are some other differences. We're still doing analyses to see if we can identify morphological characters at a species level. All right, the third solution that we've been working on is to develop a photo guide. And so this photo guide would be something that initially when I started planning out, just going to be 1 and page 2, I was working with some NOAA collaborators, and they were working on a different photo guide for a subset of the reefs in the Gulf of Mexico. And I actually liked my plan so much that they incorporated page 1 and page 2 in their layout and their technical report that came out in 2020. But the idea is that you could go to the first page and then just print every 4th page. Okay, here's my mini guide for taking on a cruise with me for the video surveys. So the first page would be in situ and ex situ images, not needing microscopy. Page 2 would be images for when we need to do microscopic work. Page 3 would be for your genetic data, and page 4 would be for that other data. If you want ecosystem-based management, so you environmental data, maybe some additional omics data. So this is what the layout would look like. You have, or what it does look like is we're building it. You have your in situ image, an image that's sufficiently clear to see the diagnostic characters and realistic of what you might actually see on a video screen. And then your ex situ image. Over there, again, so you can see the diagnostic characters, listing the diagnostic characters that you can see in these images that you can use to assign species. A comparison to similar species in the Gulf of Mexico or in that region that you're working on, and then your recommended field assignment. So if you can see this character, yes, you can assign it to genus level. But if you cannot see these characters, play on the safe side, assign it to the family level. And so when we are doing the microscopy work, these would be light microscopy images, I mean electron microscopy images, and we need to do a little cropping so it will enlarge to see everything a little bit better still, so work in progress. But here we would be looking again at maybe what you can visually see, a little bit of what you can visually see ex situ, but much more so if you just have a dissecting microscope. Not the greatest resolution, but still something to see a little greater detail. Again, description of those diagnostic characters to look for, a comparison to the similar species in the Gulf of Mexico, and your assignment recommendations, and also some repository information. This page is a little bit in progress still, but we could put map of the mitochondrial genome, so phylogenetic trees to show where this group falls relative to the most similar species. Probably some wording down here with recommendations as well. So which markers are available and where is that repository information? So what are our next and future steps? And so these are two of the genetic markers that we have. I have 5 or 6. Phylogenetic trees, and I've color-coded. You don't even know a lot about this, just each of these clades represents a species or taxa, a group, and the colored ones are the ones we're primarily going to be focusing on first, and that we've started working on. The ones in black, either someone else I'm aware of is already working on this group, so we want to work with them instead of repeating what they're already doing, or I don't have enough samples yet to start working on those. But we're going to continue to collect this data, the morphological data, and then we're going to actually start revising the taxonomy, work with collaborators, so we can update the descriptions, describe some of these potential new species, continue filling this data gap, put this guide out there, and then plan to update it regularly as we obtain additional information. So what are our broader impacts? Well, I'm hoping that we're improving the accuracy and consistency in species assignments, which means that that data that's publicly available is going to be more sound. Hopefully it'll help resolve this issue when you go to look for a genetic sequence in GenBank and it's assigned to some species and to come back and line up with all your data and you're like, where did they get this ID from, right? Want to make sure everything's in repositories. We're going to be increasing the amount of reference data so that we can use some of these more advanced techniques like eDNA and actually understand what these communities are based on the data, and just instead of saying, oh, there's some corals there. Great. We knew there were corals there. Which ones? We're going to improve our understanding of octocoral biodiversity, ecology, and function. So a lot of what we're starting to work on now is microbial community analysis that I'm trying to go over today. And then we're going to add in more data so we have a more integrated ecosystem management approach and better experience. So with that, I want to thank everyone that provided samples, helped in the field, and then these are the collaborators that Val Wright should be cooperative in this work.
So if we have time for questions, if you want to ask any questions to Dr. Easton. Good.
Yes.
Thanks for the great talk, and I know it's going to be a great resource along the way. So for getting the genetic data out, sometimes they have the barcode of life, but I think it's for a certain limited area of the mitochondrial genome. Is there a way that you're thinking that your complete mitochondrial DNA might be available? Is there like a website or?
Yeah, so some of them have been published. We basically, I think we're going to start depositing them. We need to do some more QA/QC on a few of them. We've published 2 or 3 of them so far, and so they're mitogenome reports. I also have the detailed my methods and all the scripts I use on there as well. So I'm hopeful with them in the coming year that they'll all be out there, and I'll be depositing the GenBank. So yeah, but if someone's curious, they can by all means reach out. I'm willing to do whatever looked on and share data as long as you're not. Yeah, until I just want to finish QA/QC, really. Fine.
Any further questions? Thank you.
For eDNA, is there a specific marker you think people should use if they're looking for corks?
I think they probably need to use a different marker. Yeah, because the markers that they're using, it's going to be difficult to differentiate among species. So they might only be at the genus level, in some cases family level. So what that marker is, we have been doing work on extending the barcode regions. And so using different, there we go. This slide that I pulled out because I didn't have that time is in designing new primers, we've got adjacent barcode regions that have greater percent divergences in many cases, but they have differences where there are differences elsewhere. So it is possible that they could target some of these alternative regions. So you have 100% of the 8 that we trialed with had differences and only 60-some% have differences. So using more traditional barcodes, they might only capture species differences half the time.
Any other questions for the person? Don't be shy. All right, well, I think the bridge.